Settle down, everybody. Today we're going to discuss the process of translation. Can anyone tell me what translation is? Can anyone demonstrate?
Miss Codon, I can.
Okay. And what does that mean?
It means Dr. McQuaig is the best microbiology professor on Earth. That's the translation.
Okay, well that's not exactly what I meant by translation. But this Dr. McQuaig woman sounds pretty amazing.
Can we get on with the lesson, please?
Oh, right. Sorry, Dean. Sit down, Rita.
Isn't translation how DNA is changed into proteins?
Close. The process of translation is when mRNA turns into a protein.
What the heck is mRNA?
That's messenger RNA.
Since when do you know anything about microbiology?
I know some things. I know that transcription occurs before translation. Transcription is a process of the DNA parent strand being copied into an mRNA strand. That strand then becomes a protein through the process of translation.
Wait a minute. I thought that you thought that this was translation. The point. Let's stay on topic. A DNA strand turns into an mRNA strand through the process of transcription. So let's say we have a DNA parent strand with the following nucleotides: A, G, G, C, G, G, T, A, C, A, A, T, T, A, C, A, C, T, A, G, G, and A, A, T. What will be the mRNA strand?
T,
Huh.
C?
You don't know everything. In RNA, there's a nucleotide uracil that maps to adenine. There's no such thing as thymine in RNA strands. The strand would be UCC, GCC, AUG, UUA, AUG, UGA, UCC, and UUA.
We have a parent DNA strand that turns into an mRNA strand. And then this mRNA strand pairs up 3 nucleotides to make a codon, just like this. Each individual codon then codes for a specific amino acid. Noxious way, Dean is right. Amino acids do make up proteins.
Yes, I know. And each little piece is split up by a start and a stop codon.
What is that supposed to mean?
Let me show you. AUG is the start codon. It's going to keep on going until it reaches a stop codon. Like in this strand, UAA, UAG, and UGA.
Yeah, well, did you know that this section and this section are referred to as the untranslated regions because they are loose ends that are not included in protein.
While I appreciate the enthusiasm, I'll take over the teaching now. Sit.
Oh, by the way, AUG also codes for the amino acid methionine.
Sit! Sorry, where were we? Okay, so the DNA strand codes for the mRNA strand, and then these amino acids have to get to the ribosome somehow to turn into a fully functional protein. So this little guy tRNA comes in, and he's going to come in using the A site, P site, and the E site. Aminosyl, peptidyl, and exit site.
Yeah, so since the start codon initially resides in the P site, amino acids are going to be brought into the A site while the polypeptide chain is forming in the P site.
Alright, you know what? You teach it.
The mRNA strand is read from 5' to 3', and the start codon is always AUG. Now, we have our start codon, and at the beginning, we have the A site and the P site filled with 2 tRNAs that already have their respective amino acids. And now, since these 2 sites are filled, our tRNA here in our P site will form a peptide bond to the tRNA at the A site. By doing that, this amino acid will lift this tRNA and join and start making a polypeptide chain with the tRNA number 2. right here, we'll have our chain begin forming. So now, since this tRNA has no amino acid, it will exit through the E site and leave the P site wide open. When we have the P site open, now we can allow for the tRNA in the A site to shift to this position and allow the entry of a new tRNA, and repeat the process over and over until our stop codon